The next program we have in our hand is focused on transcriptomics data analysis for chronic diseases. And in this program, we will discuss various aspects of transcriptomics data analysis, which is also called as RNA-seq data analysis, and how this specific field of bioinformatics has evolved and continue to evolve to help us understand the disease biology or systems biology from the disease biology from the systems biology aspect. So the technique and the analysis methods and the bioinformatics analysis for understanding the patterns within the data and the comparative analysis of this data has evolved and has been refined over the years. Now it is at a point where all of these data analysis can be run without or with minimal user intervention. And it is now regularly used by many scientists and is being performed, used for in-depth analysis of gene expression data and isoform expression data to study the transcriptomic landscape of the diseased state or altered state, etc. So, so we know that the genomics data has provided, genomics and transcriptomics data has provided new ways and new options and new dimensions to link targets to disease and how to validate these targets. The result of this genomics data revolution is a potential for a new era of drug discovery, and transcriptomics is at the right at the pedestal for those drug discovery of the drugs that control disease through regulating gene expression, for example, or by mimicking the mechanisms by which our body exerts control over a range of normal and disease functions. So the underlying data that is required for performing a transcriptomics data analysis, as you would have guessed, comes from next generation sequencing. And let's talk about NGS and what it represents in transcriptomics data compared with the other techniques, and also get a brief introduction to how traditionally mRNA abundance or gene expression abundance has been measured quickly. So, so as a method of measuring RNA translation, transcriptomics studies actually help us to detect mRNA expression inside the cell. And this mRNA expression will act as an accurate alternative or accurate proxy to the conditions of the cell that it represents. So this conditions and this phenotypic, this transcriptomics landscape or expression landscape can help us understand, for example, the disease biology or the development of resistance for a particular treatment, or generally the difference in the molecular pathways and difference in the biological function that one set of tissues establish, or one set of tissues exert over, compared to the other set of tissues. So at the forefront or at the basic level, we have to measure the mRNA level or the amount of mRNA present in the cells, present in the tissues. So how do we do that? Traditionally, this has been done using microarray technologies. More about this and comparing and contrasting microarrays and NGS are detailed in this program. But with each iterations or each development in technology involved in measuring the mRNA abundance, we get more and more data or more and more information regarding the cell or regarding the phenotype or disease biology of the cell. So in the end, what we would like to have is something like this, where the genes and the isoforms of the genes will actually represent the difference in the type of samples or difference in the phenotype of the samples that it is taken from. So depending upon your analysis methods, depending upon your type of question, you can either go for traditionally understanding or analyzing the gene expression landscape, or you can go for isoform expression landscape, where you will get your hands-on or where you will get information about the alternative splice forms of the genes that you are analyzing here. So this, as we said, this will extending your analysis method to include isoforms will actually help you understand or help you identify some unannotated exons or novel transcripts which have not been characterized before. So that can be very crucial in understanding the disease biology. So these patterns and gene expression patterns and transcriptomic landscape can help you identify or help you understand the biological markers or biomarkers for which is always used for used in the clinical data diagnostics and clinical therapy selection of different therapies and improvement of clinical diagnostics. So more and more of these data and more new discoveries based on these expression patterns and isoform expression patterns will help us understand these complex diseases at the subcellular level, also from the target identification for the new or repurposing of existing drugs or novel drug design. So this is transcriptomics program, and this program includes 6 or 7 interactive session. We will cover all these topics within this interactive session. In addition to this, you will get a practical hands-on where you will understand the logic behind setting up this transcriptomics data analysis, which we will discuss in detail. I will rush a little bit quickly here, and in the end of this analysis, you will get a list of output files which you can either explore them interactively or download them to use it for your report or use it in your analysis. So in addition to identifying differentially expressed genes, which is the aim of performing transcriptomics data analysis, you can also understand how different pathways are enriched or suppressed because of the genes that are differentially expressed in the conditions that we are analyzing or comparing. So the basic, this I will skip. And the outputs are, you will get a myriad or variety of outputs, which will include tables, which will include plots, and which will also include visual output, interactive visual outputs. So all of this will help you understand the biological phenomena or clinical condition of the disease. So as I mentioned, this structured data can be used for further data science based analysis, and this we already discussed, so we will skip. So you also get an introduction to single cell transcriptomics, which can help us understand all the messenger RNAs that are present in single cell resolution. So it is not like bulk cell profiling, which we have been discussing so far, where the sequence libraries are created for an averaged mRNA expression quantification for a different for a tissue. Here you will get the tissue composition at the single cell resolution. Let's quickly skip through a couple of these parts. We have one more program to cover. So this actually tells the power of single cell analysis, pipeline analysis, where you can, for example, here where you can get different cell types that populate a tissue, so that you can understand the heterogeneity behind the tissue that you are analyzing in a much more clear fashion. So this level of resolution you will not obtain, or you will not get from bulk cell RNA analysis. Bulk cell RNA analysis and single cell RNA analysis complements the bulk cell RNA understanding of the disease biology in a very upcoming and a novel fashion.